Hello, my name is Helios Leonardus. In this video, I'll be investigating mitochondrial replacement therapy, or as known as MRT, commonly referred to as the three-parent baby technique. My focus here is to analyze how this technology replaces mutated mitochondrial DNA to prevent the transmission of serious mitochondrial diseases from mother to child. In normal human cells, mitochondria are organelles responsible for producing ATP through aerobic respiration. Cellular respiration leads to an ATP production, and they occur through oxidative phosphorylation, a process that takes place on the inner mitochondrial membrane and involves the electron transport chain and ATP synthase. Unlike most organelles, mitochondria possesses their own genetic material known as mitochondrial DNA. This circular DNA molecule contains genes that encode essential protein subunits of the electron transport chain, which are crucial for efficient ATP synthesis. However, when mutations occur in mitochondrial DNA, the structure of these respiratory proteins is altered. This reduces proton gradient formation and disrupts the electron transport chain, and as a result, cells experience energy deficiency, particularly in high metabolic demand tissues. So, without genetic engineering such as mitochondrial replacement therapy, mutated mtDNA is inherited maternally and cannot be corrected. This leads to these mitochondrial diseases, which may cause internal and external problems. One important thing to note is that mitochondrial replacement therapy does not directly edit the DNA sequence. Instead, it replaces the mutated one with a healthy mitochondrial DNA from a donor egg cell. The nuclear DNA from the mother's egg, which may contain mutated mitochondria, is removed and inserted into a donor egg that also has had its own nucleus removed, but still contains healthy mitochondria. This reconstructed egg is then fertilized with the father's sperm. This method is called as maternal spindle transfer. As a result, the child inherits nuclear DNA from both parents, but mitochondrial DNA from the donor. The DNA sequence of the mutated mitochondrial genome is effectively replaced by a normal mitochondrial genome. Since the donor mitochondria contain functional genes, normal transcription and translation occur. This produces fully functional respiratory proteins required for the electron transport chain. Hence, oxidative phosphorylation proceeds efficiently, ATP production is restored, and the child develops a normal phenotype without mitochondrial disease. Based on a review article written by Hitika Sharma and colleagues in 2020, mitochondrial replacement therapy has shown promising results in preventing the transmission of mitochondrial diseases. So, basically, the article explains that techniques like maternal spindle transfer can reduce the carryover of mutated mitochondrial DNA to less than 5%. This is important because mitochondrial disease usually develops only when the mutated DNA reaches a certain threshold level. The review also reports successful births in countries such as Australia and Mexico, where babies were born without clinical signs of mitochondrial disease after the procedure. However, the article also discusses limitations. MRT does not cure existing patients. It only prevents transmission to future children. There are also concerns about small amounts of mutated mitochondrial DNA being carried over, possible mitonuclear incompatibility, and long-term safety across generations. Mitochondrial replacement therapy raises ethical concerns because it involves genetic material from three individuals. Some people question whether modifying embryos is morally acceptable and worry about how far genetic technology could go in the future. However, MRT is designed specifically to prevent serious mitochondrial diseases, not to enhance traits. In countries like United Kingdom, the procedure has been legally approved since 2015 under strict regulation and supervision. In my scientific judgment, mitochondrial replacement therapy is ethically justified. The technology prevents life-threatening genetic disorders and improves the quality of life for future children. When carefully regulated and used only for medical purposes, the benefits clearly outweigh the ethical concerns.